Now we're going to start talking about tropisms, or how plants perceive and respond to their environment. So tropisms are a way for a plant to detect a stimulus and then grow towards or away from that stimulus. The stimulus can be a variety of things. We'll talk about a few of them, with the main one being light. Let's say this is a light tropism picture here. You have, say, a light source off to the right over here somewhere, and you have these flowers that are pointing towards the light source. If you're moving towards the stimulus, if the plant is trying to get towards that stimulus, we'll call that a positive tropism. If the plant is trying to avoid the stimulus, like it is in these roots, imagine there's a light source over here, these roots would then be growing away from the light source. Is what we call a negative tropism. So a positive tropism means grow towards stimulus. Negative tropism means grow away from stimulus. So there's two different versions, and each type of tropism has those two versions to them. So we'll be talking about phototropism, a plant's ability to detect and respond to light. And we're going to spend the most time on that one. It's the most well studied, the most well understood. And their plants are photosynthetic organisms, so it's pretty critical to their survival. We'll then also talk about gravitropism, so how plants detect and respond to gravity. Also hydrotropism, so how they detect and respond to water. And thigmotropism, and how they detect and respond to touch. So we're going to start with phototropism. We're going to spend by far the most amount of time on this part. Now, if you remember grass, grass is a monocot, and since it's a monocot, it has a coleoptile protecting the shoot apical meristem from the seed, right? So it's going to emerge out, the coleoptile will be protecting it, and then as it grows, it'll pierce the coleoptile and grow into the full grass plant that you're familiar with. Now, Charles Darwin, and I think his son actually studied this, and they did a pretty cool way of trying to figure this out. But here's a little grass seedling, and it's growing up here. And what they did is they put a piece of foil over the top of it. Basically, the foil kind of mimics the coleoptile as a protective covering, but more importantly, it blocks the light. And what they found is that when they put a light source off to the right, you can see these arrows here. So there's a light source out here. Is that the seedling would grow up, but it would grow straight up, meaning it didn't seem to detect the light. It didn't show any positive phototropism or any negative phototropism. When the coleoptile was covered with the foil, the plant didn't seem to respond to light much. It did grow up. That's likely attributable to gravitropism. Now, what they did here, though, they let it grow up, and then they filled it full of sand till only the coleoptile was exposed to the light. Only the very tip was exposed to the light source. So everything down here, no light, just a very tip light. And what they found is that in that case, the plant would bend to the right, bend towards the light. So it would show positive phototropism. Now, what's really cool is that the part that would bend wasn't the part that was exposed to the light. The part that was actually doing the bending had no experience to the light, meaning that there was some way that a signal from the coleoptile was being conducted down to part of the plant, modifying its growth habits and bending it towards the light source. So that's a pretty important discovery, basically saying that plants can detect the environment around them and respond accordingly by sending signals to other parts of the plant's body. That's a pretty complex thing. We're very used to it for us, right? Like I'm moving my arm around. We know that signal is coming from my brain down through nervous system to my arms. Plants don't really have a nervous system. I mean, they do have some sort of electrical conductivity like that, but they're primarily going to be responding by using hormones. So they're going to be sending chemical messengers throughout their body and having them respond. And we will talk a lot more about hormones later on. Okay, so from here we know that the seedling exhibits positive phototropism and that it's something about the tip of the shoot that is sensitive to the light source. And that the tip of that shoot can then somehow send a signal to some other part of the body to change its behavior. But then there was another study that was similar to what Darwin did, but they took the shoot, same idea, some grass seedling here, right? But they put a mica wafer. Mica is basically a rock, it's a mineral that is impenetrable to any of the hormones or signals that the plant's sending through its body. And so what they did here is let's look at the right side here first. You have the one that shows positive phototropism, and they took that mica wafer and they put it basically halfway through the seedling. So on the side that's exposed to the light, they put it halfway through. So basically cutting off the whole right side of the plant signaling pathway. No xylem, no phloem, nothing's getting through there. And what they found is that it's still bent. it's still bent in a demonstrating positive phototropism. Now that's pretty cool because that means that mica wafer did not inhibit the signaling pathways of that plant. But what's more interesting is when they switched it, they put the mica wafer on the left side, so the side that was not being exposed to the light, the side that was dark, it was in the shaded region of the plant. When they blocked the signaling pathway, it disrupted the phototropism. So when they blocked the pathway on the left side, the dark side of the plant, phototropism no longer occurred. Now that's really important. So what that means is that whatever signal they're sending is traveling down the dark side of the plant, not the light side of the plant. So what they then did is like, well, we have an idea that maybe it's this hormone called auxin. And auxin is really a class of hormones. There's a whole bunch of different ones. The most common one is IAA. You don't have to know any of that, just know that they're auxins. That's also what we typically use for rooting powder if you ever plant propagations. But what they did is they looked at these mica wafer cuts, and they looked for how much auxin was in those regions. And they found that the shaded region had higher auxin levels than the unshaded region, indicating that auxin was probably that messenger, that the apical meristem was detecting light on the shaded region, sending auxin down its stem to influence the tissues below it, telling those tissues bend towards the light, bend to the right. So some pretty clever studies. They're trying to demonstrate how positive phototropism would work. Now here's a little diagram kind of showing that. So here's one that shows positive phototropism. Here's basically the apical meristem with a different photosystem there, the photoreceptor. Light hits it, and when it hits it, there's more auxin indicated by the bigger arrow traveling down the shaded region, the opposite side of the light. So the shaded region has more auxin traveling down it than the side that's in the light. So more auxin in the dark side than the light side. But if the light's directly above the plant, there's equal amounts of auxin going down both sides. So we know auxin seems to be the mediator of this. We know auxin is traveling down that pathway. But that doesn't explain how it actually does it. And so we're going to go into a little bit of detail here. Again, this is some pretty complex molecular biology, and we're just going to kind of scratch the surface of it. But the main idea is called the acid growth hypothesis, is that somehow there's a way for the plant cells to become more flexible and swell up. Because this phototropism can happen pretty fast. And so it's not like they're adding a bunch more cells really quickly. It's that they're dealing with the cells they already have and changing their behavior. And then over time, they will accrue more cells to change that. So the acid growth hypothesis follows several patterns here. One, we know auxin is now accumulating on the dark side of the plant. And so if you have a shoot that's growing something like this, or let's say we have a shoot that's growing straight up, if you want it to bend to the right, there's basically two things you could do. Eliminate cells on the right side or grow cells on the left side. Let me back up, I'll show you what I mean. If you want to take this shoot here, the straight stem, and bend it to the right like this, you either need to add cells over here, so that way there's more cells on the left side, basically causing it to arc, or take away cells on the right side, and basically causing it to cave in, which will also produce an arc. And the acid growth hypothesis says it's the first one. They're going to expand the left side, causing it to basically push the stem to the right. And so what they think is happening here is that auxin is going to travel down probably the phloem, down the dark side of the shoot, right? And when it gets to the cells that it's trying to target, there's a bunch of receptors there that the auxin can bind to, and it's going to trigger proton pumps. So these are active transport pumps. They're going to take protons, hydrogen plus molecules or ions, and pump them out of the cell membrane. Now, acidity is basically a measurement of hydrogen ions. The more hydrogen ions you have, the more acidic the environment is. So if you're pumping hydrogen ions out of the cell membrane, you're basically pumping them into the cell wall, which means the cell wall is becoming more and more acidic. Now, there's a bunch of proteins called enzymes that speed up chemical reactions, basically, and they often have very specific requirements. Like, for example, in our stomach, we have the protein the enzyme pepsin. It needs a very acidic environment to break down protein. Expansin is very similar. It prefers an environment that's somewhat acidic. And so when you pump out a bunch of hydrogen ions into the cell wall, expansin activates. And expansin is basically going to go down and kind of break the hydrogen bonds in the cellulose of the cell wall. Now, when you break those hydrogen bonds in the cell wall becomes more pliable. So it becomes more flexible, which is allowing the shoe to kind of start to bend over. You've increased the flexibility of one side. Now, along with that, when you break the cell wall down a little bit, basically make it more flexible, you allow more stuff to diffuse into it, particularly sugars. So more sugars start to go into the cell. With more sugars and more higher osmolarity of the cell, you've basically lowered the solute potential. So if you lower the solute potential, water is going to move into the cell. So now you have a flexi cell wall, one that is way more flexible, but then you also have more water coming in, which increases the turgor pressure. And it basically causes it to push over. It causes that phototropism effect that we saw in the previous image. So oxygen goes down, activates proton pumps. Protons get pumped out, lowers the acidity, lowers the pH, makes it more acidic. Activates expansin, which increases the flexibility, which allows more solutes to diffuse in, which allows more water to diffuse in, which increases the turgor pressure. Turgor pressure plus flexible causes it to push over. That's the acid growth hypothesis, which is mediated by oxygen. Now, that's in stems. In roots, oxygen has a different role. Very similar role, but, well, kind of opposite role. But roots don't want to grow up towards light most of the time. They usually want to grow down, away from the light. So shoots typically exhibit positive phototropism. Roots exhibit negative phototropism. They want to grow out of the light. They want to grow away from it. So in roots, oxygen actually has the opposite role. Rather than causing cell elongation and flexibility, roots actually inhibit that oxygen inhibits that in roots. So in stems, promote elongation and flexibility. In roots, inhibit elongation and flexibility. And it kind of makes sense. Think about it. If you have a root system, just like you had the shoot system that we just talked about, and auxins develop on the dark side, right? It inhibits growth and flexibility, which means it's going to cause it to turn downwards. It's going to cause it to turn in a similar fashion to what we just saw, but the opposite direction. So it's growing it away from the light source. Now we also have gravitropism. Gravitropism is detecting gravity and responding towards it. Now, if you look outside, most plants grow up. So what that means is that they have a positive gravitropism, at least for the shoots. They're going to grow up, right? And there's several reasons for that, right? Closer to the light, actually, and escape competitors, get away from the soil where there's maybe more herbivores, all kinds of other things that might be there. And so they want to get away from that. They want to expose themselves to the most light, get away from competitors, and protect themselves from whatever might be wandering on the ground. Now, what they've done, though, is they've grown seedlings in the dark and in the light. And you can see that a plant can grow up just from phototropism. The sun is above us, so the plants grow up. And that's true. If you grow seedlings in light, they grow up. But if you block them from the light, they usually still grow up. And so there is no light stimulus there. But there is a gravity stimulus. And that gravity stimulus is gravitropism. The plants are detecting that sense of gravity, and in plants, they're growing up in response to it, right? And in roots, they're going to be growing down, away from it. So auxins still play a role in gravitropism, but it's slightly different. So if you remember amyloplast, it's a type of plastid often used as a storage molecule. There's a specialized amyloplast called a statolith. Now, these statoliths basically act as like a light switch. If you've ever worked on like an old car, underneath your hood, you'll have a little light source. And when you lift your hood up, the light turns on. And when you put your hood down, the light turns off. That's because there's little liquid mercury in there. It's called a mercury tilt switch. And when you tilt it up, mercury connects to conductors and turns the light on. And when you tilt the hood down, it no longer connects it and turns the light off. It's similar in function here. Inside the little cell, the amyloplasts, due to gravity, are going to fall to different parts of the cell based on how the orientation of the root. So you can see that down here. Imagine gravity is going straight down on the slide here. All the amyloplasts fall to the bottom. Now, at the bottom of the cell here are these little receptors that detect where the amyloplasts are inside of the cell. Now, if we look over at B, if the root is growing straight down, the amyloplast will be down like that, and auxin will then be distributed equally on both sides of the root. Now, if the root was growing horizontally, like in this one here, right, the root's growing horizontally here, the amyloplast will start to fall to the bottoms of the cells, and when they do that, auxin starts to accumulate. So auxin will accumulate more towards the bottom than towards the top. So auxin becomes more prevalent in the bottom of the cell. Now, remember, auxin has the opposite effect in roots. Rather than promoting elongation, it inhibits it. And so it stops the growing of the cells there. And if you stop the growing of the cells on the bottom, the top's going to start to curve away. So it's going to keep growing, but it's going to force it to curve down. And so it's going to encourage it to grow downwards in the same direction as gravity, right? Now, they did the same test here where they look at auxin concentrations. If the root is growing straight, auxin's equal. If the root is growing horizontally, there's a bunch more auxin in the bottom part of the root. So auxin accumulates in the bottom in the same direction as gravity, inhibits growth, and causes roots to grow with the direction of gravity. Now, there's also hydrotropism. This one's not so well understood, often because there's other correlates, right? Hydrotropism is going to be associated with phototropism, but it's also going to be really associated with gravitropism, and so it's hard to separate all those things out. What we do know is that plants do seem to exhibit it. Roots definitely move towards where there's a water source, so there is a hydrotropic effect. We do know that it's not controlled the same way. Auxin does not play as big of a role in hydrotropism as it does in gravitropism or phototropism. We know it plays a role, but we don't know exactly the nuances between it. So for hydrotropism, we're just going to talk mostly about that it exists, that positive hydrotropism would be growing towards water, negative hydrotropism would be growing away from water. We also have thigmotropism. Thigmotropism means responding to touch. So positive thigmotropism, something touches like a tendril and it's going to grow towards it. Negative would be you encounter something and you move away from it, right? So tendrils, plants that climb up, they often have positive thigmotropism. They want to find objects in their environment, cling onto them, and then continue to grow up it. They're using them to climb, right? But things like roots, where they're going through soil, right? Maybe they encounter a big rock. You don't want it to keep growing towards that rock, right? That's not beneficial to the plant in any way. What you want it to do is move away from the rock, go find more nutrients and find water. So roots often exhibit negative phototropism. Shoots can also exhibit negative phototropism. It really kind of varies on the plant's ecology. Tendrils themselves just typically have positive phototropism. I can kind of show you that here. So here's a twining plant of some sort. And you can see how it wraps around and keeps going in circles until that apical meristem contacts one of these sticks. See how it contacted it? Once it contacts it, the apical meristem is pretty sensitive to touch, and it'll immediately start to wrap around it. Now, it's a more complicated story here than in gravitropism or phototropism. There are auxins at play, but there's also things like jasmonic acid at play and other hormones here that we'll talk more about during the hormone week than we will now. But now you just need to know thigmotropism is a response to touch, and that it can be positive or negative depending on the ecology of the plant. Now, a variety of plants demonstrate what's called thigmonastic movement. Basically, movement that is a response to touch that invokes a movement that is regardless of the stimulus direction. Like in thigmotropism, we just talked about with the twining plant, it detects the branch and then it wraps around it. So it is sensitive to the direction of the touch. This movement is not sensitive to the direction of touch. So think, for example, a Venus flytrap. It has like basically a mouth to it, right? Fly lands in it. It doesn't matter where you touch on the Venus flytrap, its response is close, right? That's a thigmonastic movement, just movement in response to touch regardless of the direction of the stimulus. Another one is sensitive plant. You can see they touch it here. I think it takes them a couple times on this one. And what you're going to notice is all the leaves close up. So you can see each leaf, all the leaflets are curling up on themselves. You can see how let me back it up here. Oop. If you look here, just out of the way, all of these leaves are open, and we go to the end. If I can get the hand out of the way. Leaves closed. This leaf is closing. They're kind of shading them, so it's hard to pause the video there. Oh, you can see that leaf is now fully closed. It doesn't matter how they touch it. You can actually just shake these plants, and they will all close up. There's also a common plant here locally called the bush monkey flower, and it has a stigma that has like 2 lips to it. And when you touch either of the lips, it doesn't matter which one, it closes. And it's a way to stop from, to be able to control their pollination. So thigmonastic movement is thigmotropism, but less specific. But it's also usually much faster. Okay, so to recap, we've talked about various types of tropism, photo, gravity, hydro, and thigmo. And we've also talked a lot about auxin.